I mean, we really haven't identified kind of like a cholesterol-like biomarker that can one-to-one to say, okay,
Yeah.
you're going to develop, very, very high risk of cardiovascular disease. And not only that, it's a causal, and you can actually treat the biomarker instead, right?
Yeah.
So we know nowhere near that. So that's the point I want to get through, is why the cardiovascular field is so much more advanced than we are in the cancer field, is because we have not identified that type of impactful biomarker.
Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe. Hello and welcome to BioTalk Unzipped, where our mission is to unzip, unlock, and uncover the stories behind medical progress. I'm your host, Gregory Austin. I'm a life science professional in bioanalysis in Kansas City. And along with me from North Carolina is my friend and co-host, Dr. Chad Briscoe. Chad is a bioanalytical scientific leader and, quite frankly, just a fun guy to be around. Here at BioTalk Unzipped, we bring experts from different corners of the pharmaceutical, biotech, and medical device worlds to share their innovations, insights, career journeys, and personal stories to the show. So please do subscribe, and thank you for listening. Today on BioTalk Unzipped, we are joined by Dr. Bob Liu. Bob is a senior principal scientist at Genentech Roche in bioanalytical sciences, where he develops and implements clinical assays that track immunogenicity for T-cell bispecifics and allogeneic CAR T-cell therapies. Over the past 14 years, he's supported drug development across multiple oncology companies, working hands-on with modalities like antibody drug conjugates, bispecific cell therapies, and he's been in the trenches from early discovery all the way to the clinic. Today, we're asking a question about something that I think a lot of us ask, but really not many people answer very precisely. Why haven't we cured cancer yet? So that's what we're going to talk about today. Bob, thank you for being here. Welcome to the show.
Yeah. Thank you for having me. It's quite a joy to discuss
It's
this
our
topic.
pleasure.
Yeah.
I appreciate you approaching me at the conference and volunteering to join us. So it's great to have you. So it's our tradition, Bob, to make a $100 donation in our guest's name to the charity of their choice. And you've chosen AACR, which is the American Association for Cancer Research. Why is this particularly important to you?
Yeah, AACR serves as a central hub for all resources regarding cancer topics and cancer treatment. So they provide information, up-to-date information for patients, for drug developer, for advocates, and then they also coordinate annual conferences for us who are doing cancer research and cancer drug development. So it really serves as the central resource and hub to coordinate our efforts in fight against cancer. So it's definitely one of my dear to heart
Absolutely.
agencies out there. Yeah.
Just a quick one. We are very excited to share that this episode is brought to you by our founding sponsor, Lucentra. Inspired by science and empowered by IT, Lucentra works with life science and healthcare organizations to make technology work the way it should. They help teams evaluate, implement, and get real value from IT solutions that support innovation, not slow it down. So if you're unsure about your current technology or you just want a second opinion, Lucentra is just a call away. I know John Lord. He's a founder of Lucentra, really great guy, and experienced, very authentic professional, and a good guy to work with that you can count on. So thank you, Lucentra, for sponsoring this episode. So, Bob, when people hear, why haven't we cured cancer yet? What is the first misconception you'd like to dismantle?
Yeah, we have this kind of joke. I mean, we always laugh about, a lot of people is like, maybe this conspiracy theorist think, oh, there's already a cure to cancer, but they just don't
Uh-huh.
want to make it widely available to everyone. They want to keep it selective. Maybe it's a government thing, or maybe it's like
Uh-huh.
these greedy biofarmers that you have to pay an absorbent amount of money in order to get access to these, innovative treatment. So I just want to say that's definitely not the case because you can
Uh-huh.
just take a like example such as like Steve Jobs, right? We all know Steve Jobs passed away of cancer in 2011, and Steve Jobs is one of our most successful entrepreneur and highly respected, a multi-billionaire, right? I mean, what kind of resource he cannot access
Uh-huh.
with his his influence, right? Yet
Right.
he still passed away due to cancer. So definitely not the case. And we'll just say, money can't buy cancer cures. And that's why we continue to struggle with this topic. And it's a topic that I'm highly interested. Yeah.
Yeah,
Yeah, it's
I'm
a lot
really
of it's sorry, Gregory.
no.
Yeah, it's many years of research. We certainly need the money to fund the research, right? But it's many years of research, hard work from thousands, if not hundreds of thousands of people trying to put the effort forward to cure cancer, one type of cancer at a time. That's
Yeah.
the other
Yeah.
challenge, right? There's many
Exactly.
types of cancer.
Exactly.
Right. Yeah.
Exactly.
Some are easier, obviously, to tackle than others. I think
Right.
we've had a lot more success in the bloodborne leukemias and other types of cancers than, of course, brain cancers, because we'll discuss some of those obstacles to reaching those, including the blood-brain barrier. But I'm really glad you brought this up, and we mentioned, I think, in our very first episode, that science is not a conspiracy theory. It's science. We're doing the best that we can, and you're dealing with. It's not this foreign disease that we'll tackle and get into today. It's your own body basically malfunctioning in ways that's causing these disease conditions. So Bob, you've worked across ADCs and the T cells, bispecifics, and CAR Ts. Which modality most changed how you think about what curing cancer might even mean?
Yeah. So let's step back just a little bit and discuss. I mean, let's define what cure cancer means to
Yeah.
at least the medical definition.
Yeah,
so
yeah.
so we have an idea. So these days, I mean, throughout my career, the oncologists are very hesitant to use the C word, right? The cure word,
Uh-huh.
because we all know cancer is extremely difficult to treat and it's very, very optimistic, very, very resourceful, and it always can come back and relapse for those patients. So it's a very scary disease. So, but these days, the oncologists define cure to cancer as if the cancer is in complete remission for 5 years. Okay.
Uh-huh.
Up to 5 years is considered cured, at
Hmm.
least for that type of cancer. But we
Yeah.
all know it could still come back after 5 years, but that is currently the medical definition. So, with that in mind, we can discuss which of these modalities has the higher potential as a single agent to give you this 5 year cancer free durability of treatment. Okay. So, I have worked with many, many different types of treatment modalities. My personal opinion is those modalities that can uh, involve the immune system or elicit the immune response have much better and more durable response than other reagents like just generic chemo, just like ADCs. We'll discuss a little bit in depth. So with that said, the highest potential, in my opinion, are like these immune elicitators such as the checkpoint inhibitors,
Hmm.
um, as well as the CAR Ts.
So,
They have the best potential as a single agent to give you this 5 year durable response.
so
The,
Bob, is it fair to say that that's because you're teaching your body to fight the cancer
exactly.
itself?
Yeah, yeah. So that's one concept I really want, our audience and the folks out there to realize is cancer is constantly adapting and constantly adjusting to your treatment. And our immune system is adaptive immune system, right? That's part of our immune response. So we really need to take advantage of this adaptation to continuously fight new arising cancers, mutated cancers, readjusted cancers toward your current treatment. And the immune system probably does a better surveillance and better job at keeping the cancer down than any other treatment that we have available currently. Okay.
Makes sense. And you say, cancer is a collection of diseases. What is the most useful way to categorize cancers for
Yeah.
normal humans? Is it, tissue of origin, genetics, treatment biomarkers? What do you think?
Yeah, excellent question. So, from a patient's point of view, from a non-science patient that went into the clinic and find problematic, think that the first thing they want to know is what kind of treatment are you going to give me, right?
Mhm.
What's there available for what can I, get placed on? So, cancers can definitely be placed into treatment categories. So let's take breast cancers as a paradigm. So breast cancers in the clinic is categorized into estrogen receptor positive, progesterone receptor positive, HER2 receptor positive. Why is that? Because if you are diagnosed with one of these, we know exactly
Mhm.
what treatment regimen you need to be on, right? So if it's estrogen receptor positive and relies on estrogen for its growth and propagation of the cancer, then we have a slew of estrogen receptors and aromatase inhibitors and a lot of newly developed recently degraders and stuff that's coming on board to inhibit estrogen response and cut off that circuit that cancer need. If it's HER2 positive, there are a slew of HER2 positive, a specific treatment to stop that necessity for the cancer. So you can see that by categorizing by treatment is obviously a very straightforward thing for patient and for folks that are not involving science. Now, for drug developers, we obviously are very interested in the molecular biology, the genetics, the pathology, and mainly
Mhm.
because we really want to interrogate and see if we can find, you know, other ways to inhibit cancer, find other Achilles heels that we can generate, a safe, effective drug against these cancers. So we continuously learn from these sequencing effort and molecular effort to categorizing these cancers. And also based on these molecular signatures, we can also say this type of drug correlates with better response. And these type of cancers with this molecular signature respond better to another type of drug. So we can very quickly and effectively uh, place the patient into these treatment quickly and not wasting their time on treatment that isn't going to yield very durable or beneficial uh, uh, treatment for those folks. So these are all, the molecular level is very important for
Mhm.
us as drug developers and cancer researchers. Um, so in the future, you'll see cancer is becoming more and more as personalized medicine
Mhm.
because cancer is so different, like Chad just mentioned.
Hmm.
The Consortium has categorized at least 200 different cancers. So we really need to be very specific when we say treating certain type of cancer. It has to be very
Mhm.
specific based on the morphology, the pathology, and then to give the more personalized treatment regimen.
So I have a question about, and this may be more of a diagnostic question because this is coming from my personal experience. How do you feel about the validity of methylation profiling in terms of trying to figure out what type of cancer a patient might have?
You're talking about specifically methylation profile?
But there's more. I mean, you have pathology, histopathology, you have, and then you have methylation profile, which is some people look at it as sort of a black box of trying to figure out,
Yep.
what's the the genetic makeup that may point towards, and sometimes the pathologist and the methylation profile don't necessarily agree.
Yeah,
I'm just
yeah. So
curious what you think
methylation is
about that.
just one biological tool you can count as one level of biomarkers. So cancers and development certainly have these, development have very set methylation patterns to prevent, to say these are more highly proliferative, more regenerative stem cell-like cells, and then these are more differentiated cells. And methylation certainly play a great role in that. And cancer co-opt some of those mechanisms so they can remain more stem-like and have high proliferative potential. So methylation is not probably in terms of value of contributing to molecular signature is not any more than like genetic signature or RNA profiling or
Mhm.
sequencing, right? It's just a different level. But all these signatures can help us design a molecular signature that says this type of cancer respond to this type of drug and this type of signature respond. So that's kind of our overall goal. I mean, we all continue to evolve and continue to learn. I think there's still a lot of learning, but I wouldn't over advertise like methylation signature is more important and more relevant than any other signature because I think for me, they're all valuable. We just need to find that specific signature for
Right.
certain type of cancer and provide the correct regimen of treatment.
Because in my experience with my son's low grade glioma, it we really struggled to get a definitive diagnosis. Is it a DNET?
Mm,
Is it pilocytic astrocytoma?
mm,
What does that mean for treatment? And it really came down to that genetic signature.
I see.
And what's really interesting is you can have a DNET that does have an FGFR1 alteration, which is our son's case. It's a complex
Mm,
DNET, or it may not. And that genetic identifier will really dictate treatment for him, especially if there's a targeted therapy
mm.
that comes to pass. And there's a few experimental ones that are out there. So I just have some personal, experience with that.
Yeah, yeah, and
It,
certain
it,
cancer probably those are weigh a little bit more because more known about it, and other cancers rely on other type of signature. Yeah, sorry. Go ahead.
Bob is an expert in cancer, and Gregory is unfortunately somebody who's done a lot of research because of your specific situation with your son. If there's anyone listening to this who just wants to learn about cancer, maybe they've been diagnosed, maybe they're finding themselves in your situation, Gregory, what are resources that people can go to, reliable resources that the common patient, right? Somebody who's doesn't have the scientific background as we do. Where could they go to get good information and help themselves with their conversations with their physicians? So I think either, Bob, maybe you start,
Yeah,
and Gregory, I'm sure you
sure.
have an
Yeah.
opinion as
Yeah.
well, because you come
Sure.
from a totally different perspective.
Right.
Yeah, so
Yes.
I mean, for me, I think I mean, as a researcher, we have to look at all the standard care currently for that type of cancer. And then where I look for standard care, there's a
Mhm.
specific website. Maybe we can put a link in
Yeah.
eventually in our talk. I'll send you guys the
Yes.
link for that one. And then AACR again is a very valuable resource,
Okay.
and you can go in there. But yeah, for very relatively rare cancer, the resource might be a little bit more limited
hmm
versus the other more common cancers. Yeah, from AACR. Yeah, both are very valuable resource.
Yeah, Gregory, what was your experience trying to travel this road?
Yeah, so we searched broad and wide, and we really employed AI to kind of help us find the literature out there that for a DNET, which is diazoembrioplastic neuroepithelial tumor, which is supposedly benign. It's not really. It's still a low grade glioma that can grow and cause some issues. Or was it pilocytic astrocytoma? And the reason I asked that methylation question is that the pathologist kind of overwhelmingly said it's a DNET or a complex DNET. The methylation profiling came back as 98% pilocytic astrocytoma. So we're like, what is it? Because what we thought is one of those labels will drive the treatment. DNET, it's either surgery or a radiation, pilocytic astrocytoma. You can add chemo to that, but DNETs don't really respond to chemo. And so what we did is basically a broad lit search on what we could get our hands on. And one of the standards of care for that, of course, is GTR, gross total resection, which unfortunately wasn't accomplished in the first two surgeries. That's the best standard of care you can get, and that usually can be curative for kids. So we're very lucky that he has a low grade, slow growth type of a tumor. But it's either that or radiation, and we don't want to give an 11-year-old radiation therapy at all, to be honest. And maybe if he's older, if it comes to that, we could consider it. But too much radiation can actually cause high-grade gliomas, which is something we found in the literature as well. But that's really just as much of the literature we could find and reading and summarizing and kind of using AI to help us find it faster and summarize it quicker is what was helpful, regardless of the actual source it was pulling from. We made sure they were, legit articles, but they were, interesting articles from all over the world. I mean, there was a number of cases like in Saudi Arabia where they were studying DNETs, which was interesting that maybe local doctors don't know as much about. So that's kind of how we tackled it and what we found out. And then we dug in, once you have some labels, that's where you can kind of dig in and do deeper dives, which is what we did.
Yeah. Oh, that's fantastic. And I think, we definitely want to put that link because, boy, we want to keep people off TikTok to try and figure out their cancer treatment, right? So
100%. Well, I think it is time for us to move into our next segment, New News.
New News.
New news.
Oh, I love it. Well done, Bob.
In Regulatory Focus of RAPS on December 9th of 2025, the article FDA urges randomized trials for CAR T approvals reports a significant shift in the FDA regulatory expectations for cell therapy development. So the senior leaders at FDA Center for Biologics Evaluations Research, CBER, announced the future CAR T approvals will generally require randomized controlled trials rather than the single-arm studies that supported many first-generation approvals. While the agency acknowledged that 7 CAR T products have already been approved across 18 indications using single-arm data, the FDA officials emphasized that this approach should now be the exception, not the rule, reserved for rare, heavily pre-treated diseases. So going forward, sponsors will be expected to include appropriate control arms, clearly justify any equivalence or non-inferiority designs, and then engage early on with the FDA for that endpoint selection. The FDA also highlighted the growing importance of biomarkers, but cautioned that these must be well validated if they are to serve as surrogate endpoints. So, Bob, the FDA mentioned this potential use of biomarkers when randomized trials aren't practical. From your perspective, what does it actually take for an immunogenicity or other cellular biomarker to be credible enough to influence regulatory decisions?
Yeah, I mean, I think for cancer is still, the gold standard is still overall survival. That's a gold standard for approval. But we have these interim readouts such as PFS, progression-free survival, and these things.
Mm-hmm.
Yeah, I mean, we'd love to have some biomarkers that can give you a much better sense of, you can achieve this kind of overall survival. But currently we don't have, across the board, we don't have that, those. What we do have is we have some biomarkers to say your cells are active, and those are easy to track of. Okay, so what FDA said about, having a control arm, we definitely struggle with that here at Roche Genentech, and that adds tremendous amount of cost to that because, mainly because specific to the cell therapy. So for all the other, for like bispecific, for ADCs and for other, yes, we're gonna add in the standard care or the other control arm, right? And that's actually pretty feasible to do. Here, the cost for so let's say for NHL, right? So non-Hodgkin's lymphoma, the CD19. So one of the standard care is CAR T. So you're going to have to go head to head against a CAR T, which is fine normally. However, the cost is so prohibitive to do that comparator arm, mainly because, the cost for CAR T these days is around 600,000 per treatment, right? So, so each patient you enroll in that control arm, you're looking at 600,000. And how many patients can you really enroll in that control arm to justify the
Right.
cost? The cost, right? So that's very, very cost prohibitive, specific for cell therapy. I mean, for bispecific and for others, it's not as bad, and we certainly have those control arms. So we've been going back for our CAR T program or a specific CAR T program and argue back with the FDA to say, another standard care for our program is T cell bispecific. Can we use T cell bispecific instead of the approved autologous CAR? Save us at least like 200 to 300,000 per patient for this treatment. And so we're going back and forth with FDA on that.
Yeah, because it's already expensive enough for those treatments without adding to that. I know there's,
Yep.
FDA is all about safety, and we certainly appreciate that, but there's kind of always that battle between industry and FDA, between safety versus innovation, and where do you draw that line and what's practical and reasonable. Seems like, just from my opinion, that's, not very practical for autologous CAR Ts.
Yeah, it's very cost prohibitive. You can see that. So,
with an already very expensive treatment.
Yeah.
yeah,
Interesting decision.
exactly.
So, Bob, you shared some slides with us earlier on just to kind of prep us for this conversation, and I appreciate that. I wanted to share one because I was reviewing it, and it was some very interesting data, and I'm a visual person, so I'm going to go ahead and share this on the screen here. So, so what we're looking at is detection, diagnosis of cancer at a stage where mortality rate can or can't be impacted. And what I found very interesting, so we've got you're looking at the mortality rate grid here. You've got heart disease on the left, you've got cancer on the right. And you see in the heart disease, it's going down quite a bit for the higher income. These are separated by levels of income of people. I'm curious what the intent was of separating people by what their income is. But what's interesting to me is, and you see, of course, cancer is just going up. We're from 2000 to 2015, we're making some headway in heart disease on the higher income. Everything else is going up. The two things I found interesting is there's higher incidences per 100,000 of deaths in the higher income, then regardless of which way the line is going, than in the lower or mid income. Because lower is, it's really like, cancer is pretty low comparatively to the higher incomes. I don't know. What do you think about that data?
Yeah, how do you interpret these data? Yeah.
What do you think about that? I found that really interesting.
Yeah, it's very complex data, and I mean, the point that I was trying to make. Let's go back a little bit and give a little bit more context. So what I want to make a point is that cancer treatment is a lot harder than cardiovascular, and the
Yes.
treatment options and the treatment advances is not at the level of the cardiovascular. So if you just Google what is the number one cause of death in US, it's still cardiovascular, followed by cancer. However, that rate of death increase per year over year, for heart diseases, it's flattening out. So on the left side, there's the simpler graph. You can see that rate of increase, it's starting to flat out. Whereas cancer in that blue line, it continues to go up.
Continual rise.
So what it does say is we have made tremendous impact on the mortality due to cardiovascular over the past 20 years. whereas we have not made nearly as significant of impact on cancer. So let's break down a little bit. And then in terms of income, so this another level of complexity is that
Right.
if you look at the higher income, you assume those people have the resources, they have the education, they have the knowledge of what kind of state-of-art treatment and preventions and diagnostic out there, right? So they have this access to resource. So those folks that can have access to these cardiovascular diagnostics and treatment, they're doing quite well. And you can see the death rate is going down, dropping off a cliff over the past 15, 20 years, right? And
Yeah.
continue to go down. Whereas, yeah, with the same resources for the high income earners, they have the same resources and all those. The cancer treatment access or the diagnostic is nowhere have the same impact
Mhm.
on cancer, right? You can see cancer continue to rise no matter how much money you make. And I just give that
Right.
example of Steve
Right.
Jobs,
Right.
right?
Yeah.
So, and then when you look at lower income, you can assume they have less resources and less access to these treatment, and therefore, their mortality rate is still increasing. Now, let's dive a little bit further into the molecular pathology of these diseases. So, cardiovascular, we know, is a much simpler disease. It's basically a drainage or like a blood flow issue, right?
Yep.
So, I mean, if your artery clogs, it increases chance for these cardiovascular diseases. And then the major advance in this field is that discovery of cholesterol, namely the low density lipoprotein, right? LDL. So
Mhm.
now we all know that LDL is bad because it can form plaques, it can cause clogs in your blood vessel, and then that can lead to higher chances of developing these cardiovascular diseases, right? And then the really major impact is the cholesterol is not only a great biomarker to identify your risk, it's also a causal marker, a
Mhm.
driver of cause of this disease, right? So now you have these treatment like Lipitor that actually treat not heart diseases. You don't even have heart disease at that stage yet. You just have high cholesterol that might lead to heart diseases down the road. But now you're treating cholesterol with these Lipitor and stuff, and to decrease those and dramatically decrease heart disease issue. Again, like heart disease and cancer, if you catch it early, if the person have actually heart attack, it's much more acute and much dire, right? You really have to, you have a very short treatment window to revive the patient. But if you can prevent it, right? If you can find this biomarker, not only a biomarker, a causal biomarker that actually contribute to the progression of the disease, you can dramatically impact the mortality rate for
Mhm.
that disease. Now, we have no such a thing for cancer, but we kind of expect that because cancer is a very complex, this collection of diseases, right? You're not going to expect one thing to be a causal. So
So
that's why we have
I
not found that
Mhm.
Bob,
gold standard.
if we were to normalize that data against total number of diagnoses, do you think it would have a similar trend? Or, for instance, are wealthy people getting cancer more frequently now? Are they being diagnosed more frequently versus poor? I think it'd be really interesting to normalize
Mhm.
that data that way.
Yeah, yeah.
Yeah, no, it's
Good
a
point.
excellent point. I mean, I'm a big proponent of early diagnostics because my
Mhm,
postdoc research is for early diagnostics.
mhm,
And if you can catch cancer early on, before it metastasizes, right?
mhm,
I mean, surgery and radiation, it's very, very advanced. You probably don't have a major issue if you can catch it early and you
mhm,
surgically remove or followed by localized radiation. Most people will not die from cancer if you catch it that early.
Mhm.
However, catch some type of cancer early, it's extremely difficult. We just don't have those biomarkers. The best thing is it's the imaging, like the CT, MRI imaging, right? but those have limitations. It's not going to catch very small cancer,
Hmm.
and cancer at very small stages can already metastasize. Just depends on the progression, that type of cancer, right? That molecular
Hmm.
signature. Um, so that makes it very, very difficult for certain cancer. While for other cancers, that's why we are very big proponent. Go do your colonoscopy when you get
Hmm.
to that age.
Yeah.
Go
Yeah.
do
Yeah.
your this
Yeah.
imaging, do your mammogram, why? Because if you can catch it early, it's not as big a issue, right? Now, it also gets back to the healthcare issue. And this, I think, what you'll see very big divergence between the different countries. Here in US, right, you go do a physical and you go do a checkup. Okay, I want to check. They're like, what, you can check cancer?
Hmm.
Hmm.
Like, well,
That's
what biomarker you're going to check? Or you're
Yeah.
going to do it, I was just in China like 3 months ago, and my wife dragged me to do a routine physical, and this is routine. They do it annually, and usually the workplace sponsor these annual checkup. So just from the blood work, there's like at least 10 to 15 very famous, very well-known cancer biomarkers, and they check that routinely. And they give you CT and MRI and X-ray and ultrasound, part of the annual checkup.
wow.
so, so we can see maybe in the future this divergence of early diagnostic and catch of cancer in other countries versus U.S. and North America, mainly because driven by this healthcare system, and a lot of are
Hmm.
not willing to do these diagnostics. Of course, it's a balance, right? I mean, you don't want to give people random X-rays, especially young developing people,
Hmm.
right? Because we know the risk of X-rays and stuff. But yeah, it's a balance. I mean, maybe young people don't need X-rays like every year. That I think that's over treating, but maybe every 5 years, every 10 years, we can discuss those, based on studies and data. But definitely early diagnostic can have an impact on cancer tremendously, but we have to implement that.
So
Wait.
one of the diagnostics that I've seen recently, and there's a number of different companies that are doing something similar to this, but a company Gregory and I have talked to a couple of times called Toby Health, and Toby, what they're doing is urine screens and then doing headspace analysis, looking at a profile of everything, and they don't even have complete diagnostic information, or they don't have definitive identification of what all these peaks are, but they're running it with GCMS, so it's non-specific. It becomes a pattern recognition and an algorithm and a math problem. Have you seen that, and what do you think the future is for not just Toby, but that general class of
Right.
pattern recognition, non-specific identification approaches?
Yeah, I mean, it all come down to the detail. I mean, as we study these gene signatures, the issue is we still don't have like really, really faithful biomarker or set of gene signature for
Hmm.
every cancer, right?
Hmm.
Certain cancers a little bit better, and certain biomarkers we know very well, like
Yeah.
BRCA, we know very, very well, like
Hmm.
people who have both copies of BRCA mutated have about 80% chance, greater than 80% chance of developing breast cancer when they reach
Hmm.
age of 40. So yeah, but these biomarkers are far and few, right? I mean, we
Hmm.
really haven't identified kind of like a cholesterol-like biomarker that can one-to-one to say, okay,
Yeah.
Hmm.
you're going to develop, high, very, very high risk of cardiovascular diseases.
Yeah.
And not only that, it's a causal, and you can actually treat the biomarker
Yeah. Yeah.
instead, right?
Yeah.
So we know nowhere near that. So that's the point I want to get through, is why the cardiovascular field is so much more advanced than we are in the cancer field, is because we have not identified that type of impactful biomarker or gene
Yeah.
signature yet.
Yeah.
So, what does it mean when you say we can't eliminate every cancer cell?
Yeah.
What does that mean practically? Is it a biological limit? Is it a technology limit? Is it both?
Yeah. Yeah. So, yeah, excellent question. So, one of the major struggle as a drug developer against for cancer treatment is whatever drug we give to to treat cancer, we just subject the cancer, those multi clones, various clones that co-evolve into the selection process, right? It's just selection for whatever can survive through your treatment, and that'll become the predominant clone. And that clone likely to relapse and cause the it'll be the predominant, you know, cancer that rearises following the treatment. So this goes for chemo. This goes for all sort of current treatment we employ. It's just a selection process, right? So
Yeah.
that so that's where the challenge of achieving durability is. So, okay, so I want to take you guys to this analogy of antibiotics.
Hmm.
So we have this analogy in our field. If you look at antibiotic, and oh, they employ antibiotic to treat bacteria infection, right? It's actually extremely difficult to get rid of every single bacterium in an infected person, right? But what
Yeah.
the antibiotic can do is greatly slow down the progression of the bacteria, right? To allow our immune system to catch up and to develop, to mount our immune response against the bacteria, right? So that is extremely important. So we really need to elicit the immune system to get rid of all the last bacterium. If you don't elicit the immune system, it's unlikely you will
Hmm.
get rid of every single bacterium. So
Right.
we take the same analogy into cancer. Whatever treatment you're giving right now, other than the ones I'm talking about, why I really a big proponent of pushing for the immune related treatment because we must engage the immune system, allow the immune system to be the surveillance, to be the active, adaptive monitoring and treatment of cancer. As the cancer has evolved, our adaptive immune system will also evolve accordingly to keep us suppressed. And we all know, if our immune system is weakened due to whatever
Mhm.
reasons, these opportunistic infection diseases come right back, right? So that's why the immune system plays such a big role in infectious diseases, and you also need to play the same important integral role in cancer treatment to give us the durability of treatment, right? So that's the concept I really want everyone to be aware of.
So I've heard that your body is generating cancer cells maybe on a regular, semi-regular basis, but
Yep,
you would never know it because your immune system is taking care
exactly.
of those cells
Yep,
also on a regular basis. So you don't notice
yep,
it until you become symptomatic in some way.
yep.
But is it, I don't know, is it more of an immune problem or is it a uh, genetic mutation problem causing these cells to become cancerous and do things we don't want them to do, or is it a combination of both
It's all
breaking
of them.
down?
It's all of them. It's. I mean, so cancer is so resourceful, right? I mean, like you said, we probably are generating cancers constantly in our body. But if we have a very, capable immune system that can recognize those cancer cells, recognize the differences between the cancer cells and the normal cells and get rid of them effectively, yeah, you don't ever see the problem. The problem arises when you're more immune compromised and you or cancer adopt a way to hide from the immune system, such as they downregulate MHCs. So now they don't present any peptides up. That's one very common way for them to downregulate the HOAs, and now they escape cancer, escape the immune system, immune surveillance. And then there are many other concepts. It's just this hot and cold tumor. What we mean is the hot tumors will have a lot of immune infiltrates, which
Mhm.
means there's a lot of, you can take an analogy of there are a lot of cops there to catch any robbery or any theft or any crime that's occurring there, right? But then you have these cold tumors, which lack of these immune infiltrates, lack of T cells, and they're lacking, cop or authority surveillance, right? So how do we convert these cold tumors into hot tumors? How do we, activate this re-recognition of these cancer? That is the major topic that we're engaging with all these immuno-oncology effort, right? So really elicit engaging the immune system to recognize it, to do
Right.
something about it, and to really suppress the cancer. Yeah, but it's a push and pull mechanism because cancer will continue to adapt new ways to escape the immune system. Because if they don't escape, they will not survive, right? So there's this huge selection pressure for them to adapt, but our adaptive, our immune system is also adaptive. So it's this push and pull and just this continuum. Yeah.
So Bob, changing gears a little bit, you're a bioanalytical scientist, I think would you would classify yourself as, and all of all these new types of treatments and diversity of modalities and everything has really expanded the field. Can you talk a little bit about how you navigate which tools you use in the different modalities and just
Yeah.
help us understand the complexity of being a bioanalytical scientist in
Yeah.
today's world versus
Yeah.
25 years ago when it was LCMS and maybe an immunoassay.
Yeah. I mean, to be honest, I'm relatively new to the bioanalytical world. I
Hmm.
spent, majority of my career in discovery research. I just joined Genentech
Mm,
a little over 3 years ago to get
okay.
into the bioanalytical world. And I really do enjoy it. I love the clinical side, supporting
Yeah.
the clinical side. But yes, there are a lot of challenges, new challenges for bioanalytical world. Mainly a lot of these new modalities, right? And the immunogenicity has become more and more upfront and challenging for the bioanalytical field and how to monitor immunogenicity. How, but for me, because I didn't come from bioanalytical field, I'm kind of not just satisfied with monitoring immunogenicity. What can we do about it? That's what I
Yeah.
care.
Yeah.
How can we, yeah, well, how can we mitigate, right? I always ask that question.
Right.
Hmm. Hmm.
So we also taking some effort in some this mitigation effort, right? But in terms of monitoring, such as these new modalities like CAR-Ts, like these newly designed bispecific molecules were highly unnatural, right? I mean, before when we talk about these more traditional antibodies, especially with humanization,
Hmm.
the immunogenicity is largely not a huge problem unless it's for certain
Right.
modality that stimulate the immune system and that those things, right? But now it's up and center with all these new modality, and immunogenicity might be the number one hurdle for a lot of these new modality. So namely gene therapy, right? So we know they deliver gene therapy with a virus, and the reason you can only give it once, just like the last speaker you guys had in the last
Yeah.
video.
Mhm.
We talk about why you can only give it once, because if you elicit an immune response, it's just ineffective to give it again. Okay.
Right.
Same with CAR-Ts. If CAR-Ts can build an immune cellular response to the CAR-Ts, you can give it again. You will not expand. It will be eliminated by the immune system. And then now we can get into even more challenging for allogenic CAR-Ts. Okay, that's even more challenging. And the last speaker talked about that a little bit. But that's what we're struggling with currently with my current project. And the allogenic CAR-Ts are another level of immunogenicity because you have
Mhm.
this allo reaction, which
Hmm.
is just very, very similar to organ transplant,
Yeah.
right?
Yeah.
Mhm.
So why do
Mhm.
you need to select organ? Why you need to match MHC and match HLA? These kind
Yeah.
of things.
Yeah.
It's the same issue with allogenic. So allogenic is at this minute, it's nowhere near the potential of autologous CAR-Ts. Just
Hmm.
put it in perspective, autologous CAR-Ts, why it has durability? Because, the CAR-T is known to expand at least you can easily detect these cells in the patient for at least 6 months. And some extreme cases, you can detect up to 15 years. And they still have these cells, these
Wow.
CAR-T cells.
Yeah.
up to 15
Hmm.
years, right? Or
Wow.
even beyond.
Yeah.
Okay.
That's
And
In the
outstanding.
allogenic,
yep.
in the
Hmm.
allogenic field, okay, let's switch to that. What's the best you can get it to? 28 days. We're talking about a month
Really?
compared to
Yeah.
15
Hmm.
years. Well,
Wow.
six
Wow.
months
Wow.
is 6 months is the standard we're trying to achieve. We're
Yeah.
nowhere
Mm.
close to
Yeah.
that. Okay, we're
Wow.
trying to a lot of different. Why is that? Immunogenicity, rejection by our immune system. Okay, and
Okay.
this is really an allo rejection
Yeah.
issue. Um, yeah.
Okay.
So that's yeah.
Oh, that's fantastic. I appreciate that
Yeah.
perspective, Bob. And just for our listeners, the previous guest that you were referring to was Christopher Hopkins, CEO
Mhm.
from Glafabra. So just so
Mhm.
folks can look that up, that was also a fascinating and highly related
Fantastic.
discussion. Yeah, yeah, it's amazing what they're doing and the complexities that they're trying to solve at
Yes.
that company. Yeah,
Yeah. Yeah.
yeah.
Wishing him the best and success with that. And also for our listeners, most of you already know this, but autologous is your own cells and allogenic would be somebody else's cells. And that's why you have that immunogenicity and the rejection issue going on there. And I appreciate, Bob, how you open things up, that trying to dispel, the general public and their conspiracy theories about, oh, there's already a cure for cancer and they're just suppressing it so they can make money on treatments. But then you look at how difficult it is to identify first diagnosed cancer while you can still do something about it before it gets to be a real problem, and then actually try and treat it in many different ways that we're trying to treat cancers. If you could give patients like one honest statement that is hopeful but not hype, what would that be?
Yeah, I mean, over the years, I mean, progress is very slow when we in the trenches. But if you look back, when we declared fight and war on cancer in the 1960s,
Hmm.
we have made tremendous progress on certain cancers for sure. So when I was doing my postdoc days, I really thought we would not see a cure to cancer in my lifetime. But, the immuno-oncology field changed that dramatically. And certain cancers are absolutely cured. And then these physicians can say, those people who are on Keytruda, been treated for a year on Keytruda, and we wean them off that drug. And now 5, 7 years later, we detect no cancer for those
That's
people,
amazing.
right?
Yeah.
And I met patient who are stage 4, declared that you should just go to hospice.
Yeah.
In fact, this is actually one of my real estate agent. He's declared stage 4, and he's supposed to go to hospice. But when he was placed on Keytruda, he's completely cured, and he's working as a real estate agent and just going back to his normal life, just like that, right? And then you hear President Carter have cancer that metastasized into his brain, and he's cured because of this immuno-oncology.
Hmm.
So we're very, very hopeful that certain cancers can be cured, and definitely we see that, right? So the challenge right now is how do we expand that to bigger population of people? The unfortunate statistic for immuno-oncology is about 20% of the folks respond really well to
Hmm.
these immuno-oncologies. And those people, once they respond, and they usually have very durable response
Hmm.
and usually can be considered cured, but only 20%. So what about the other 80%, right? So
Yeah.
that's what the fields continue to try to do something about it. We talk about why certain cancers don't respond because they don't have immune cell infiltrate that they're considered cold tumors. They don't have T cells there already to employ and attack the cancers. So how do we convert these uh, cold tumors into hot tumors so that these immune oncology drugs can work, right? So these are the active research and active new modality drugs that are coming online. But I'm very, very hopeful. It's a great time to be in this cancer research
Hmm.
field, right? And with such a fast pace in new development and stuff. But I also want to step back and tell everyone that, cure to cancer is a milestone. It's an endeavor that's going to rival with any challenges or achievements in humankind, right? It can rival with any past challenges, like we learn how to fly, we go to the moon, go to Mars, go to doing interstellar travel, or rival with any future imaginable achievement, like faster than light travel, or
Oh,
for
Yeah.
engaging
Star
aliens,
Trek.
right,
Yeah.
anything you can imagine,
Yeah.
because cure to cancer is so daunting, and we discussed some of
Uh-huh.
these topics today, right?
Mhm.
And cancer is just so diverse. It's 200 different diseases at least, right?
Mhm.
And we are moving toward this personalized medicine and providing a specific regimen for specific patient. So the immune system we're talking about, we can cure cancer. So that's one dogma and one side of treatment. Another theory, another thing we're trying to employ is if we have enough of these treatment, such as chemo, such as ADC, such as bispecific, they might not last as long as if you engage the immune system. But if I consider these as all tools in a toolbox for treatment regimen, right? If we have enough these tools and we can regimen a certain different type of tools to implement for certain cancer, we might be able to manage the cancer and suppress the cancer very similarly to how we manage like chronic diseases like diabetes, like high blood pressure.
Yeah.
And maybe cancer isn't such a problem if we have a huge collection of these tools and we can employ the tools at different time and give different effect. And if we have gene signatures to predict when to use certain tools, right? I definitely think that's very likely to happen. Is we can do these combination treatment regimen to really manage the cancer to a level that maybe we can convert it to a chronic disease and not such a acute death, scary
Mhm.
disease.
Mhm.
Yeah.
Yeah.
Now, I'm optimistic as well. Hopeful. I'm certainly hopeful for my kiddo that some of these targeted therapies come along in case it comes back, because you never know. You just don't.
Mhm.
Yep.
Mhm.
And we can do the best that we can do and try and take care of our bodies in the meantime, so we're not doing those things that might increase our chances, right, from having cancer. Bob, thank you so much for reaching out to me, coming on the show, having this important conversation about what has been done, why hasn't been cured, what we can do, and where we're headed. Appreciate that. It's great to have you on the show, and thank you very much for your time.
Yeah.
And
Yeah.
Bob, yeah, thanks for me as well. We could have this conversation all day. I think,
Yeah. Yeah.
thank you for your fight against cancer. We often thank our war heroes, and I think we should, but I think sometimes there's other people in this world that we should thank that are fighting and saving lives and working so hard every day. So thank you for being on the front lines of cancer research and development.
Yeah. Thank
Indeed.
you for having me. It's a pleasure. If I can have like one more advertisement.
Please,
Go
please.
for it. Yeah.
So,
Anything.
yeah, I mean, I just want to say to those folks who are interested in getting into the field, especially those young, you know, um, talented, driven, dedicated folks, we definitely need your help. We need
Mm.
more folks to get into the field to continue this fight against cancer, right? I mean, I'm doing what I can currently, right? But we need
Uh-huh.
to continue enlist these new blood and new perspective and new talented people to continue this. So, folks who are interested in cancer, folks who are interested in discussing career development in our field, feel free to reach out to me. I'm happy to address any questions and help with you getting
Yeah.
into this field.
Mhm.
And Bob, what are the best
Yeah.
avenues to reach you?
Mhm.
You can send me an email, and yeah, I can. You can link my email, my
Sure.
Sure.
personal email, which is
Right down below.
boblu77, yeah, gmail dot com, in the link. Yeah, I'm happy to address any of these questions.
Fantastic. Thanks, Bob.
Yeah, thank you.
Yeah.
You know, it's funny because we, like you mentioned, Chad, we celebrate heroes, we celebrate sports heroes, but you know, there's no Super Bowl, there's no World Series for medical science or
Yes.
really any science. You know, we need to celebrate those people who are making dramatic differences in people's lives, and why we're here in this world is because the type of work we can do can dramatically affect somebody's life. So thank you for that.
Yep.
And that's why I really appreciate this kind of form. I mean, I think we can get the word out there, can get the conversation started, you know, to engage people. And I'm a big fan of what you guys are doing. And
Thank you.
yeah.
Thanks, Bob.
And
Appreciate it.